Hello, everyone. Before starting, I would like to thank all the organizers for actually being persistent and putting in the effort to make this happen. So far, for me, it's been a very enjoyable experience, and my only goal for now is that it continues to be so. My name is Martin, and I'm a PhD student in Delft, the Netherlands, where I work on a project where we attempt to develop a model system for the bottom-up reconstitution of chromosome organization. We call it the Genome-in-a-box. See if I can move forward. Yes. So DNA is a fascinating molecule, and we don't seem to be able to learn enough about it because we've been looking into it for over 150 years by now. It was in 1865 that Austrian/Czech monk has understood that there are certain factors that contribute to expression of traits and thus heredity. And it wasn't until 80 years afterwards that we understood that DNA is actually responsible for carrying these factors. In very Pauline line of research, a Swiss biochemist, Friedrich Miescher, working in Germany, was the first person to isolate DNA in 1869. And it wasn't until about 8 years afterwards when we understood what is the molecular structure of DNA by concentrated effort of many scientists. This led pathway for us to appreciate the complexity of various processes that DNA engages in, because on its own, it's a passive molecule. And just one of our hallmarks is the discovery and description of the central dogma. But now we understand that the regulation of the expression of genes is an involved process, and DNA engages in it as much as proteins that associate with it. So there is more than 200 transcription factors in E. coli and more than 2,000 in eukaryotes. So it's really huge ensemble of proteins. And the systems that we are attempting to study are characterized by complexity. They are very robust, partly possibly because there is a lot of redundancy in quantitative systems. Getting a bit more zooming in onto the 3D genomics aspect of what we are talking about, DNA is hierarchically organized in both prokaryotes, eukaryotes, and also in archaea. So on the smallest scale, it's wrapped and bent and looped by proteins that interact with it on molecular scale. And further, it interacts or builds chromosome interaction domains, topological associated domains, and yet further forms domains of self-interactions that have been described in eukaryotes as A and B compartments. And on similar level, there are macrodomains, that those are regions that tend to self-interact and stick together in prokaryotes. We are all in the same business, and that is to try to understand how this works, how it's possible that the 1,000-fold or even 100,000-fold compacted DNA in prokaryotes and eukaryotes fit into the volume of the cell and can be tightly spatially and temporally regulated, such as to allow a regulated expression of genes. So there is basically a pathway to research, right? And we, with our project Genome-in-a-box, we are really zooming here on the controlled interactions. So we all are trying to create mechanistic descriptions of what is going on. Some of us have huge amounts of data that they can make some sort of unsupervised inference on. We corroborate our results with simulations. We attempt to create predictive models. Thus, these predictive models allow us to make controlled interactions. And if we are very good in making predictive models, the control interaction is what we want it to be. And eventually, we can think about applications. In this moment, we are very much looping in this cycle. And I would like to talk about how we can have very defined interactions in a model system. So then I would like to present you the idea of the genome-in-a-box, where we start from a bacterial cell, take out the DNA, so burst the cell open, take out the DNA, strip the DNA of all binding proteins, thus obtain a megabase scaffold, and then we associate it sort of ad libitum, one by one, with some building blocks of chromosome organization. And they can come from any kingdom of life. It can be structural proteins, it can be more of active processes, and it can be also more of physical phenomena like crowding or confinement. So how do we go around about obtaining this megabase scaffold? We start from an E. coli cell. We blow it up, fragilize its cell wall, embed it into agarose plaque, lyse the cell, remove all the binding proteins from the DNA, label the DNA with a DNA binding dye, melt the plaque, and thus obtain our scaffold for experiments, which is the megabase scale DNA. This kind of approach is heavily relying or heavily more inspired by what has been done for genome transplantation in mycoplasma. So now I would like to talk about two example applications that one can think about doing in the genome-in-a-box kind of setup. So first, I would like to talk about the structuring proteins or structural proteins, in particular nucleoid-associated proteins that are known to be present in prokaryotes and structure the chromosome. Here I am showing you two most abundant ones, FIS, which is the most abundant in exponential phase, and DPS, which is the most abundant in stationary phase. So we start our experiment with isolated DNA that adopts this expanded, let's say, structure and is very mobile and sort of fluffy, one could say. After adding FIS protein, we see that there is local maxima or local clustering of compaction, and the whole object moves more like a rigid body, whereas now it's very, very flexible body. This is somewhat corroborated by the AFM experiments that we've also conducted, where you can see that the FIS has ability to bridge DNA by forming multivalent interactions among its monomers, but also binding to DNA. In similar fashion, you can observe that DNA is very much compacted by adding DPS, which is known to condense nucleoids in stationary phase. Strikingly or interestingly, it seems to retain a lot of the dynamicity, the object that we obtain after addition of DPS. And this is interesting, especially in context of recent experiments where it was shown that addition of DPS or DPS as such, DPS condensed nucleoids, will remain very accessible for transcription machinery. So it's somewhat puzzling that such strikingly condensed objects still can be very accessible, and possibly the flexibility that is retained may be crucial for that. Next, we are also looking into options how to extract more material physical properties of the DNA scaffold we are looking at. To do so, we have a system where we have two spots on the DNA, as is illustrated here, at various distances from each other. This is basically done by LECO or TETO or frost-like spot arrays. And we can track the motion of these spots in time and extract the mean square displacements, but also values like correlations or anti-correlations of movement and velocity. Next, I would like to talk about experiments that have been conducted in our lab by Roman Barth, who was a master student at that time. So very much in the spirit of what we do in our lab, we took a DNA scaffold and a loop extruding protein, as was shown that these proteins have the ability to loop extrude. And we combined this megabase scale scaffold with these proteins. So you can here already see what is going on. Over the course of about 15 minutes, we observe compaction of the DNA, which we quantify by the radius of gyration, which is sort of a weighted sum of the mass of the monomers. We also observe that there is distinct change of material properties, whereas in the beginning, the object seems to start in an expanded and very flexible state. Over time, it adopts more of a state of a rigid body and moves around rather rigidly. So far, the observations are rather phenomenological, so we are working on quantifying it even further. And in the next slide, there is one example of such quantification, where you may have already noted that on the previous slide, there are clusters emerging in the condensing body of the nucleoid. So here we indeed quantify that there is an increasing DNA content, which we identify just as a fluorescent intensity per cluster. And we observe that the number of clusters increases and they are persistent compared to control. Thinking further, we are interested in combining the isolated DNA with some microfluidics techniques. In particular, we would be curious in putting two genomes, each labeled in a different color, into a liposome, which we will have ability to squeeze into confinement And by doing so, we are interested in testing this theoretical prediction of entropic segregation of two circular polymers in confinement. With this, I would like to thank all people involved in the project, especially Anthony, who was spearheading the project, Case and other people involved, and the whole CD Lab who provides really encouraging and nice learning environment. And with this, I would also thank you for your attention and take any questions you may have. Thank you.
Thank you, Martin. I will start with questions. Very, very nice visualization technique. So for the case of the bacterial chromatin, so you showed us results with these FIS protein.
Mm-hmm.
So you compare actually the structuring of the nucleoid, where you have visualization, and AFM, but I guess AFM, that the results that you presented are on a plasmid.
Indeed, those results were on plasmid.
Okay, so the scales are very different. So that was my question: is how many clusters, how many proteins within a cluster in your nucleoid, and does it relate to your results on a plasmid? And also on a plasmid, is there some sequence effect? Okay, many questions. Sorry.
Thank you very much. It was actually a really nice question, and thank you for it. I cannot give you an answer at the moment because these experiments were rather preliminary, and we are still developing the assay. We are sort of making sure that things are happening roughly as we expect after addition of a protein, but we are not yet in the moment of really making this quantification of the number of proteins per cluster. I do agree with you that this will be interesting, and we will certainly look into it.
So we have a question by Ilya Flymer, who asks, okay, does the DNA remain circular in this experiment?
Yeah, again, very good question. So DNA is very fragile, and even more so when deproteinated. So what we have done to sort of assure the number of monomers we have and we are looking at is that we do calibration to the fluorescent intensity. So we are able to look at the object and be able to say, this is likely fragmented. It seems to be about 2 million base pairs. We can look at the object and say, this is likely fragmented, or this is likely not fragmented. It's about 5 million, 4.6 million base pairs. We are not able to say if the DNA is circular or not at the moment.
Okay. So actually, she had a follow-up question, which was, did you compare actually? I mean, you can linearize the chromosome actually in these these bacteria, which is, I mean, they work and they grow fine actually. So have you looked at that, or do you plan to
No,
look
this
at
is
that?
certainly an experiment we would like to do. We would like to designally cut the chromosome, make sure it's linear, then and compare whether we can quantify differences between the chromosomes that have been cut on purpose and those that have been not. But we haven't done it.
Okay. Okay. So Mathieu Lavigne, who asks, do you have RNA in your experiments?
Not at the moment. The idea is start from a really pure scaffold, but we would be interested in multiple things, and one of them is adding RNA as a process that may inject in supercoiling, and it may perhaps loop out certain regions of the chromosome. But we are also interested in adding RNA transcription as a readout of our technique from some, let's say, probe genes on the chromosome.
Okay. So I think I got back to a question which was related to your loop extrusion experiment. So the results that you showed, I think, are neck DNA, isn't it?
They are, once again.
It's performed on pure DNA, naked DNA, no protein, no histones, no
So
whatever on it.
It doesn't have histones, but it has the nucleoid-associated proteins associated with it.
Okay, so
Not
these
entirely
are
pure. It still has some bacterial nucleoid-associated proteins associated.
okay. So the loop extrusion was done with the SMC from bacteria.
from yeast. It condensing from yeast.
Okay, so this is a condensing from yeast performed on the
Bacterial chromosome.
okay, on the bacterial chromosome. So, and is there any results, preliminary result, not published with the SMC or and
So the effect was ATP dependent. Is interesting in the context of recent papers where there has been shown also SMC ATP independent function. And then we are looking into quantifying the speed at which this compaction happens, and then perhaps more fine resolving the final structure of the object, because we end up having some sort of local minima of clustering, these clusters that emerge. It doesn't all merge into a single block. So we would like to better understand that.
okay. But no preliminary results on the bacterial condensing, let's say.
No.
Okay.